I want to take the opportunity this morning to tell you a little bit about the NHLBI Next Gen studies and the incredible wealth of cell line materials. Sorry, I'm fighting the end of a cough, and it's chosen right now to kind of spark up. So there's just a tremendous wealth of not only materials but data that will be associated with these cell lines that I want to make you aware of that could be incredibly useful in your research programs. For those of you unfamiliar, the Next Gen project, or the Next Generation Genetic Association Studies, is a 5-year, $80 million project funded by the US NIH National Heart, Lung, and Blood Institute. And the purpose of this study was to investigate functional genetic variation in humans and the effect that it has on disease phenotype across a very diverse set of patients. 9 independent programs were funded, some of the most elite labs in the country, including those at Boston University, Harvard, Johns Hopkins, Medical College of Wisconsin in collaboration with CDI out of Madison, Scripps Research, 2 programs out of Stanford, UC San Diego, and Penn. Together, the groups are doing what they call disease in a dish. They're generating cell lines that will serve as surrogates for disease models, for disease phenotypes that you'll be able to use in vitro and in vivo investigations. A total of more than 1,500 patient-derived iPS cell lines initiating from either blood or fibroblast tissue have been already derived in these laboratories, and they cover a wide variety of heart, lung, and blood-related disorders, including cardiac arrhythmia, coronary artery disease, diabetes, issues including insulin resistance, various different heart malfunctions, cholesterol, including genetic-related high cholesterol issues, lung QT, platelet thrombosis, hypertension, a nice collection of sickle cell anemia cell lines, sudden cardiac death, and a wealth of other types of disease profiles, all focused around the heart, lung, and blood area. The most important thing about these cell lines is the data that comes with them. So not only are the disease lines materials available, but the group of 9 consortium members decided on a uniform panel of characterization to be performed across the line, which includes marker expression, differentiation into specific lineages and into 3 lineages. Also, they've done identity testing to match back to the primary material, which seems kind of like a no-brainer, but you'd be amazed how often it isn't done. And very important, as well as a tremendous amount of genomic data, including whole genome sequencing across a number of the lines. And included with this has been done on a very diverse set of patients, which is the first time I personally have seen this much diversity in any kind of collection. But it also includes a very diverse collection of controls. So looking at sex, age, and ethnicity, there will be a very nice panel of somewhere between 40 and 60 cell lines that will be normal, healthy controls as part of this collection for use. So I'm very excited to announce that WiCell has been named as the exclusive external distributor of these cell lines. So both the cell lines and the data for the cell lines will be available through WiCell. The data will be freely available to academic, nonprofit, and commercial as well, and easily viewable or linked to from the WiCell website. We are just now starting to collect these cell lines and get them in-house. We made a trip to San Diego last month to collect the first about 250 lines, and we'll be making these all available on the website soon. So finding the lines is going to be the key, and finding the data, accessing the data that goes with them. And to enable that, WiCell is in the process of updating our website to include a new intuitive cell line search tool. We're in the final beta testing right now, and it should launch right after the new year. So be able to go into this page on the WiCell website and use the drop-down menu to insert any of your parameters of interest. So for the particular parameters that are of primary interest to you would be the ethnicity, sex, gender, disease model, anything like that. You can insert those. And then when you hit the search bar, it returns to you out of the 1,500. Well, there's 1,500 in this collection. There's about 2,000 in the overall WiCell collection at this point. The cell lines within the collection that match your criteria, so you don't have to spend a lifetime searching through them. And it gives you a much more manageable list. So once you do the primary search, you'll see the search results table come up. From the search results table, we tried to put the most relevant information to help you narrow down your list to be able to quickly access the data you're interested in. The dbGaP link will be available directly from this page. You can link directly into dbGaP, sign in with your credentials, and access the whole genome sequence data from here. Or if you click on the cell line itself, it'll take you even a little bit deeper into a table that includes a tremendous amount of information and more links to external information on the cell line. So for any particular cell line, we'll have detailed cell line information, including the cell name, any alias that's been used in publication, and that will be searchable, so you can search from the pubs directly. Any diagnostic information and the phenotype, whether it's a carrier, whether it's affected, whether it's a familial partner. The familial relationships aren't represented in this table, but they will be represented. Links to both affected and non-affected blood relatives, age, ethnicity, where the tissue origin is, the method in which the cell line was derived, and then also links to external sites to get additional information directly back to the provider. For those of you who are commercial organizations that may be interested in accessing the material, that's where you would go to for appropriate licenses to license any of the disease model lines. Links directly into dbGaP, to Eagle Eye, which is, for those of you unfamiliar, a Harvard-based data repository, which will contain additional data information on the cell lines. You can link directly into there. HIPSREG is the European registry, and for any of you who are using these cell lines in Europe, we are making a valiant effort to get all of them registered in HIPSREG to make them easily usable with EC funding. And then also any PubMed abstract that's been published on them, and then additional information on other cell lines in the collection. In the current information line down at the bottom, there's a link to the PDF for the current lot we would be distributing. That would contain all of the characterization reports, easily viewable, and links, information on passage number, links to protocols that are used to culture the cells, and additional information like that. So again, all of the data will be, you won't have to create an account to log in. You'll be able to easily access all the data. If all you're interested in the data, you can get it right off the website. If you're interested in the cell lines, or you're an academic and nonprofit group, they can be ordered directly from here. Commercial groups contact the providers for information. That's all the information I have. I want to make sure that I acknowledge the groups specifically that are involved in generating these materials. They are all actively involved in research, and my understanding is that there will be publications on the majority of these lines coming out within the next 6 months. Early Brickell out of Medical College of Wisconsin, Lou Becker and his team at Johns Hopkins, Chad Cowan and the group at Harvard, Kelly Frazier at UCSD, Tom Kortemus and Marlene Rubinovich, both out of Stanford, Daniel Rader, working at Penn, Marty Steinberg and his group at Boston University, Eric Topol out of Scripps. Also, Richard Pierce at Eagle Eye has done a lot to get this data easily available to the researchers. I can answer any questions on the collection if you like, or did you want to move to the next speaker?
Yes, that's the next question. Everybody hear that?
So the question is, are they genuinely patient-derived in the moment, or are they previously patient-derived stored in research, commercial research banks like Coriell or ATCC or banks like that? We're collecting the data right now, and we've got most of the information from all of the providers on the origin of the materials, and from what I can see, they are universally patient-derived. I haven't seen any come through from Coriell. And there are MTAs that research, when you order the cell line, you'll be required to sign an MTA, and it's specific to the cell line. Some of them have specific restrictions for areas of use. Most of them do not. So if you're in, there are some that are restricted to cardiac disease-related disorders, and some of them are not because of the age. There's a nice variation. Some of the donors are quite young, so for some of the younger donors, there are additional restrictions on how those materials can be used, but it's very nice to see that kind of diversity in the collection. So,
Neil, you were saying that there are publications coming out, but that is not a requirement that people have published information on these lines, or no, they can just provide them and put them in the bank and then make them available.
Yeah, I'll do my WiCell pitch. For any of you who have a cell line, and you're looking for a distributor for that line, please contact me, info@wicell.org, and it'll find its way to me. But we do receive research-derived materials for distribution, particularly if you're going to publish on them. It's a requirement to make them available to the academic community, so we're happy to assist you in that. We don't require publication on the materials as we receive them. And for a lot of these lines that are coming in the 1,500 plus, not all of them will be included in publication from a particular group. 2 or 3 representative lines probably will be included in the publication, but we make a wide variety of the clones available.
And you're providing information on the lot number. There must be a particular reason why you're providing that information.
You know, as we all know, there's a lot of variation, not only from cell line to cell line, but there can be a tremendous amount of variation, lot to lot and passage to passage. So at WiCell, we do repeat characterization on every lot that we distribute. We always repeat identity, karyotype, mycoplasma, and sterility. So you'll know when the material comes from us that it's clean and it's stable, at least when it leaves our facility. And we do track the passage number, so you'll know how low and how high the material is, and be able to track its changes in it over time.
Great.
The clinical information that's provided is going to be dependent on the provider and what they collected and what they are based on the MTA, based on the donor consent at liberty to provide. Some of this will link to a significant amount of clinical data because, and I didn't mention this, but should have, one of these studies contains the Framingham heart patients. So there's a tremendous amount of clinical data associated with those that will be accessible. The clinical data won't be stored at WiCell, but we will provide links for you to be able to get back to where it is, where the provider has stored that information. But particularly the Framingham heart study has a wealth of information, if you're familiar with that, to get back to the clinical data. Always we put the diagnosis on, and the only diagnosis is that we will both include clinical diagnosis and then self-reported, but we make a point of separating them. So we not only report primary diagnosis, but comorbidities as well. So if you're searching the original link page, you may end up with a comorbidity that's self-reported, and you'll be able to identify that on the website.
Great to be here and to see a very nice turnout on the last day of the meeting. And so I want to tell you a bit about modeling neuromuscular diseases, and I want to specifically focus on the muscular aspect of that. And compared to many other disease areas, like in the neurodegeneration field or heart disease field, this is an area which has been a bit neglected, mostly due to the difficulties of obtaining skeletal muscle from pluripotent stem cells. So just a very brief introduction. So our main interest is on neuromuscular disease therapeutics discovery, and we use stem cell technologies to do that. And those are very long-term goals and projects. So in the short term, we also commercialize our technologies through the sale of a number of research services, mostly to biotech and pharma companies, and particularly to academic scientists. Our media and our cell lines are also available. And we originated in Sydney, Australia, and Steve already mentioned, expanded into the United States recently and opened up a lab in San Diego. And our history is somewhat unique in the sense that we were essentially a spin-out from a very successful IVF clinic in Australia. So the Genea group is now 3 companies, fertility for IVF treatment, biomedics for medical devices, and Biocells for the biotechnology and stem cell work. And the company has been around for a long time, so the IVF business started in 1985. So in terms of our strategy, so it's all about stem cells, chemistry, and screening. And we start with our cell bank, which is human pluripotent stem cell lines, mostly embryonic stem cell lines. And we have now just over 150 cell lines representing also about 30 genetic diseases. And those disease-affected lines are derived from pre-implantation embryos which are screened for those diseases in our clinic. And we've been in that area for quite some time, so we derived the first of those lines back in 2004. And shortly after that, we also created the first GMP human embryonic stem cell lines in the world. So we have the cell lines now, But really, to do something with those cells, you have to differentiate them to the tissue or the disease-relevant tissue. And so we developed a number of differentiation protocols using chemistry and high-throughput screening. So in our case, our main interest is in neuromuscular diseases, so it's neurons and muscle. And then depending on the disease we're studying, we think there's some diseases which should be perfectly treatable with a small molecule. So we develop a disease model and a drug screening assay and screen small molecule libraries. And then we think there's also some diseases in that field which would benefit from cell therapy. So that's another aspect, but I won't go into too much detail here today. And really important for this work has been the high throughput and high content screening, so the ability to analyze cells at high throughput and efficiently. So muscle differentiation process is a 3-step, 3-week process. And when we started off developing this a few years ago, everyone was telling us, oh, you're crazy, this is so difficult. And there were no reports of any muscle differentiation to yields other than a few percent in cultures. And the only other way to make skeletal muscle to some degree was to overexpress myogenic transcription factors, which has its own problems and limits the scalability. And so we developed our protocol, which was the first high-yield protocol, which was so efficient that it didn't require any cell sorting steps and no overexpression of myogenic transcription factors. It's a very efficient protocol. So for every pluripotent stem cell that goes into the process, we get about 150 to 200 myoblasts. So that makes it very scalable. And also importantly, if you do want to use stem cells for disease modeling and drug screening, you have to be very consistent in your quality. So every batch of cells that you produce has to be of the same parameters and quality. And you also have to compare different cell lines. So you also have to make sure that every cell line that you put into the process gives you a very similar outcome in the end. So we have validated our method with about 25 cell lines internally, and also send our differentiation media to a whole range of collaborators around the world, really, and who tested the method with their cell lines. And we haven't had a report yet of someone not being able to make muscle using this method. So when you look at the cells in the end, they form those nice long tubes, as you would expect, express all the markers like dystrophin shown here. And what is also quite interesting, and see, if you sort of watch carefully that video, you can see them twitch in culture. So it's not that they beat like cardiomyocytes, it's not synchronized, it's just sporadic twitching, and can be so strong in some cases that the cells actually dislodge from the culture dish. So one disease that we were particularly interested in was facioscapulohumeral muscular dystrophy, or FSHD in short. And it's a genetic disease which results in progressive muscle weakness. The age of onset is typically in young adults, but there are some rare childhood cases as well. And it's after Duchenne-Becker muscular dystrophy, the second most common, affecting about 1 in 7 to 1 in 8,000 people. And the mechanism is a very complex epigenetic mechanism involving a regional chromosome 4, which is called D4Z4 repeat. And so you can, to use an analogy and to make it simplified a bit, so if you imagine you have a garden house which has some kinks so the water can't come through, and you unravel that and the water flows. This is exactly what happens on the DNA here. So the DNA is compacted and through the disease-causing mutations unravels and then upregulates a gene, it's a transcription factor called DUX4, and that's toxic to skeletal muscle. I should also mention that we work quite closely and had the support of 2 foundations here, FSHD Global in Australia and Friends of FSH Research in Seattle. And just to summarize the results here, so we conducted a fairly extensive study using 9 cell lines, which included 6 embryonic stem cell lines from our own collection, And we also obtained 3 iPS lines from our collaborator Dan Miller in Seattle. And 5 of those 9 lines were FSHD affected, and 4 were normal controls. And when you differentiate those to skeletal muscle, they look superficially all the same, so they form nice myotubes. There's no obvious problems there. So when we looked a bit more carefully, first of all, we're interested in, do those cells express DUX4 as the genetic mechanism would suggest? And yes, exclusively in FSHD-affected myotubes, we find DUX4 expression. And also through a collaborator in Massachusetts, Charlie Emerson, we looked at a range of known FSHD biomarkers and transcriptional targets of the DUX4 transcription factor. And they could all be identified in those cells. But then also using image analysis, so you can stain your cells and then use sophisticated software which measures the morphology of those myotubes. And in this case, we found, analyzing thousands of cells in those dishes, that there's a significant difference in the morphology, and that FSHD myotubes are slightly thinner, they don't fuse quite as well, and they're shorter as well. We also found more proliferating cells in myotube cultures, which would suggest a differentiation defect, and it's known from other FSHD studies without using stem cells that there seems to be a differentiation delay, so that is what we think is recapitulated here. And we also analyzed the gene expression profiles of all those controls and the FSHD-affected stem cell lines. And again, you find very distinct genetic signatures of the disease-affected myotubes. And that's things like specific signaling pathways, like Wnt notch seems to be affected, cell cycle, the oxidative stress response. And we could actually validate many of those gene families directly by looking at effects at the cellular level. And also important to note is that those phenotypes that we see are DUX4 dependent. So you can reverse all those effects using siRNA directed against DUX4. So many of those parameters can be developed into bioassay that we could use for drug screening, and that's what we have done and we're working on. And so just to summarize our experience with working with stem cell-derived skeletal muscle, so we can produce it now in large quantities and importantly of consistent quality. And I always find it quite fascinating when you think about it. We make cells in a culture dish which are 4 weeks past the embryonic stage, and we're trying to look at effects of an adult onset disease. And invariably, as far as I'm aware and in our experience, we do find disease-related effects at the cellular level. So those cells are very useful to study the disease mechanism, potentially to find novel drug targets, and we are using those cultures now for drug screening for FSHD. And just to give you a bit of a future outlook, so we are ourselves working on expanding our pipeline, developing more functional tests. And one challenge with disease modeling is always the maturity of the cells that you get from in vitro differentiation, and that's a problem across the board, whether you're looking at neurons or cardiomyocytes, and that's the same with skeletal muscle. So we know that our myotubes, they're not adult like myotubes; they're at the embryonic or neonatal stage, which is fit for purpose and seems to be good enough for what we need. But one future direction is certainly that we and I'm sure others in the field are trying to make more mature cell types. And lastly, I want to show you some preliminary data from a collaboration with Dr. Don Low and Bijal Shah at Duke University, which was supported by the Huntington's Disease Society of America. And I mean, skeletal muscle is one aspect, but if you're looking at neuromuscular diseases, we shouldn't completely neglect the neuro aspect of that. And so skeletal muscle is controlled by motor neurons, and so we're working out or optimizing a method now to combine motor neuron and skeletal muscle cultures. And you can see in the graph on the right, in the middle, the red picture, that's alpha-bungrotoxin, which stains neuromuscular junction. So there's some evidence that the motor neurons and the skeletal muscle cells can interact. And as a general direction in the field, I believe that's one of the next frontiers, not to make just single cell types, but combining different cell types in a very functional way, which gives you more complexity to study diseases. And that's all I had to present. Thank you.
Thanks, Steve. Well, good morning, everyone. Thanks for being here. So I can see that the slides I show here is I represent the Bug Institute for research on aging. But as Steve mentioned in the beginning, I also have a company called WiCell. So this is actually the work that developed both by my lab at the Bug Institute and the let's see if I can, and the company I found here, WiCell, that we lease the space at the Bug Institute. So I'm just going to give you a very brief of the outline, what I think of screening is about. And I'm going to mainly focus on using the iPS-derived cells for toxicology and neuroprotective studies, as well as for monitoring disease. So this is just a very brief background on iPS technology. I'm not going to go through the details of this, just to give you a few points why I think it's important to use iPS technology for screening. As you all here, I guess you agree, it's offer an opportunity to obtain large number of the cells. And you can really direct them into different lineage, and that means you can study the disease in the same allelic background. And of course, it can be genetic engineering allow you to create reporters and knockout lines to model the disease. So all this, that means, iPS technology can incorporate all the advantage shown here. And if we can make it cost effective, then it will be a very efficient way to do the screening in a primary model. So here is the screening technology, what I think is important, at least this is my perspective, which we have been working in the past 5 years. So first of all, you have to make lines. And again, as WiCell just showed you, there are a lot of lines are available. So I'm more talking about a panel line for a focus screening, okay? So in my case, I'm going to use lines for neuroprotective and toxicity and neurodegenerative disease modeling. So again, you can generalize the idea. You make the panel of line, you need normal controls, you may need patient-specific lines and isogenic controls, and you can also make reporters. So after that, you have to be able to make differentiate cells and in a assayable format. So it's not like everything you can do in a 60 millimeter dish. So in this case, I'm going to illustrate just very briefly. We make neural stem cells, and then from there we can make neurons, astrocytes, and oligodendrocytes, all representative of the central nervous system cells. And after that, you have to be able to use them to run the screening. And then again, as I mentioned, my lab is really focused on toxicity, neuroprotective studies, and really my basic research lab is to look at the mechanism of action in focus on Parkinson's disease. So I'm going to give you some of the lines we have made, control lines, iPS lines. We have made many of them and use integration methods, for example, like the episomal vectors. And those are all generated from cord blood cells. And here I also have a list of the lines we have made. It's just a sub-list of the line we have made. As again, my lab is focused on Parkinson's disease, so we have made disease line from Parkinson's disease patients. For example, these are the lines that from patients carry various mutations. For example, PARC2, LARC, and GBA, and PINK. Okay, all these are generated by Sendai virus, and all the lines has been through vigorous characterization. That means we have been test the pluripotency, ability to differentiate, long-term stability in culture, and also, of course, the identity. And many of them we have done whole genome sequencing, and all of them we have done whole gene expression analysis. I'm happy to share those data with you. In the interest of the time, I didn't put all the data here. So the next line I want to show you is the reporter line we have made. Again, my lab is focused on neuron lineage, so we have made reporter lines in all major neuron lineage. For example, like neuron reporter, astrocyte reporter, and oligodendrocyte reporter, as well as neural stem cell reporter. And the method we use zinc finger nucleolase, talent, and more recently CRISPR to make the knock-in line. Okay, so this is one example I'm going to show you here. This is a neuron reporter here. Again, what we have done is this all are made by the knock-in in the endogenous allele, and we use MAP2 as a reporter for neurons, and we tag an analog and halotag into the endogenous MAP2, and this line is specifically made by zinc finger nucleolase. And as you see the line, as you differentiate into neurons, you see the luciferase activity increase here, okay? And just to validate the cells that the luciferase activity or the reporter, we also have a halotag that is a dual reporter here in the system that is coming from the neuron, so we have done a double label that you can see that specifically. One specific of the lines I want to mention is when we are making this line, we're actually working also with pharmaceutical companies, which we have been repeatedly told that it's important to have an assay that you can have the quantification, and that's the reason why we choose luciferase. And in this case, the luciferase is secreted into the media, so that means actually you can just take a small aliquot of the media and do the assay, and this can be non-invasive when you are looking at this. And in this case, if we look at the neurons, we need as less as 10,000 cells in the media. So if you do lyse the cells, you only need 10 cells to be able to measure the luciferase activity. So here are some of the other lines, as I mentioned. Again, we have neural reporter and astrocyte reporter and oligodendrocyte reporter. I didn't list the neural stem cell reporter here, but those are the lines we have created. And the other thing we have done is to really just to make the isogenic knockout lines to model the disease. And in this case, we knock out the genes associated with neurodegenerative disease. Again, my lab is focused on the Parkinson's disease. We make many of them. For example, we have a Parkin knockout, and this is the DJ1 and pink. All this, we also create double knockout, and this is very easy to do so now with the CRISPR-Cas9 technology. And we also made some of the knockout lines in other neurodegenerative disease. Again, all these lines are made from one control line, so all the cells are isogenic. So that we think is an advantage when you are going to run the screening. And at the same time, when we make the knockout, we also create monoallelic isogenic lines as we do 2 steps. Sometimes we do 1, okay, depending on the screening methods you do. So in case of dominant disease or recessive disease, even that, you can look at the dosage of the gene by looking at the monoallelic line. And the next slide I want to show you is just to let you know how we can scale up and make the differentiated cells using our system we have developed in the past 10 years. And here what we do is we always created an intermediate stage that's called the neural stem cells. We make neural stem cells from pluripotent stem cells, and then at this stage, we can freeze the cell down, and then they can be expanded in vitro or differentiate in vitro, depending on how you want to do it. And from here, you can make, from the common neural stem cells, you can make neurons exercise and oligodendrocytes. Again, so I think the advantage is doing this way, when you're running the screening, you can reduce differentiate time, which also means you can provide more consistencies if you start from your stem cells instead of from the iPS cells. And again, as I say, this stage of the cells can be cryopreserved, and then you can ship the cells also at that stage too. And we have many publications of this system. This is just one of them. So the last slide, this is the one I'm going to show you. So again, I'm not going to show you any visual data, but they all published, and some of them, we're writing the manuscript. I'm happy to share the data, but again, I don't have time to go through it. So I'm going to give you 3 examples. One is the toxicology assay, which we have been working with the NIEHA's, actually the company I found Excel, we have a SBI grant with them. So this is really just to use the cells I just described from a common neural stem cells. We make neurons exercise and also test with the iPS cells. And then we screen a compound library, which provided by the NIEHA's 80 compounds and run in a 96 well plate, and we look at toxicity to neurons exercise, neural stem cells, and compare to iPS cells, and this has been published, okay? So use this system I just described. And the other study we have done is to use the iPS or EAC-derived dopaminergic neurons to look at the neuroprotective effect for Parkinson's disease. Again, this paper is published. What we have done is to look at a small compound that has all of the compounds in there has been reported for neuroprotective effect in rodent model or in human immortalized cell line model. We took 44 of those compounds and then run with our iPS and human EAC-derived dopaminergic neurons, which we found is only 18 of them were protective to human-derived dopaminergic neurons. And it's not a surprise, these 18 compounds, most of them were the ones used for clinical trial. So that's just a way to tell you how we think the human primary derived dopaminergic neurons are more relevant to disease, okay? So this also has been published. And the last example I want to show you is to really look at the mechanism of action. Again, in this case, we use the combination of patient-specific parking line, which is Park 2. As I show you, we have 4 patients from there, and then combine with the isogenic cells that we create a knockout, and use the combination to look at the phenotype. And in this case, we were very lucky that we saw a phenotype in mitochondrial. With the parking, all 4 patients from Parkinson's disease patients, they have a reduced mitochondrial fractions in dopaminergic neurons. And this phenotype has been recapitalized in isogenic knockout lines. So at this point, we are just going to follow with the isogenic cells as we see all the phenotype has been repeated from the patient-specific line. Again, this paper has been published. So with this, I'm really done with the presentation part, but I'm happy to take any questions. Reporter. Yeah, we are more on the high throughput side, but also the reporter line we have, there is a HaloTag, which actually you can use the live ligand to do the high content image too. But we mostly focus on high throughput with the luciferase readout.
Question for all of you is, so all three of you are providing cell lines, different products, basically, from the things that you're doing. What would be the number one thing that if you could overcome a certain technical hurdle or other, maybe it's not technical, it's maybe some other roadblock, what would increase the use or more orders, more sales for products in your area? What are we facing today as far as getting more people to, why aren't more people using them? Or is everybody, is it easy enough to do it? There's no problems.
I think probably access to the materials is one of the easiest, and access to the data. And so I think all three of us are making that kind of information available. It's difficult as a researcher with the funding getting tighter and tighter and tighter, and the cost of experiments getting higher and higher and higher, to want to invest the money in a cell line that isn't already proven. So having publications available on cell lines that are being offered is, at least from WiCell, we see it as being essential. If a cell line hasn't been published on it, we are primarily a curator rather than a distributor.
Yeah.
And we're happy to provide that role. We think it's important, but they're not being distributed.
So somebody calls you up and says, well, I really want that line for long QT syndrome, but I have this problem. I mean, I can't generate cardiomyocytes, or I don't have the technology. What do you do? What do you tell them?
So because we focus primarily on the pluripotent, our expertise doesn't run to that. We will always connect them to the provider or the depositor. And being at the university, we have access to a large number of researchers who have expertise in that area, who might be able to offer them appropriate methods, protocols, and things like that. But one of your questions sort of went initially to what are some of the barriers to use. We're starting to see more and more GMP clinical grade materials being manufactured, and the one area that it feels like we're still missing or just now beginning to see breakthroughs on, it has to do with matrix, GMP, xeno-free, clinical quality matrices to move forward is an area that is now starting to break open a little bit, but still needs some work in that area to see the transition happen from research to clinic.
So if somebody wants to get a cell line, an iPS cell line, and you tell them where they can get the rest of the reagents for the culture and propagation of them, you'll do that.
So the protocols we provide actually have a section that includes materials, and it gives you vendor and catalog number. And sometimes it gives you options for vendor and catalog number. So, and anytime it is a replaceable material, we'll add the words "or equivalent," so you know you don't have to use this. There are a number of, we happen to buy our DF12 from Life, but DF12 is made by hundreds of groups out there, and from my experience, it works pretty much the same. So.
I just want to make a comment about you, have the standard, also publications. Actually, I didn't really talk about that. We have really been working on that perspective for all the line, and because I'm coming from academic lab, so I think it's very important that you can provide all the data. Like as I mentioned, many of them we have done whole genome sequencing, and you don't need to do for every one of them, but I think they need to have some sort of standard. So when people choose the line, they have something to choose, not only like male, female, and also the imprint information and other things. So we should have some sort of standardization. And in that effort, actually, we have just published a paper about the cGMP manufacture line. This is actually made by Lonza, but we have a follow-up paper that we have provided all the genome sequencing data and array data and like the SNP and other analyzer, just to make it as someone can compare to other lines. With that line, we chose like 10 line to compare and make that as a package for researchers to use. I think that's very important. So I will give you a perspective of my conversation with the FDA, because we are trying to move toward clinic. So we ask them, what is needed for a line to be used as a study material for clinic use. So we come with a long criteria, like for example, do we need to do whole genome sequence? Well, the answer is that we don't know, but you have to look at if you're going to use for certain, like let's say Parkinson's disease, they want to check with some of the mutations that are associated with Parkinson's disease. You can do it with SNP analyze or with other things. Basically, there was no standard right now. So what we try to see is to see if we can define some comparability assays, like saying here, this is what we see. This is the minimum requirement or whatever it is defined by the FDA for clinic use. So then we come up with that, then people can choose whether they want to do it or they think this is too much, this is too little, or there will be at least something to start with. I think that's important.
Yeah, we'll probably distinguish between the academic and the pharma markets as well. So I think for academics, I completely agree with Tenneille and Xianmin. It's mostly about awareness also, and having publications certainly helps. Or we found with our stem cell lines, for example, the embryonic stem cell lines, and in the US, there's the NIH stem cell registry, and many of our lines are registered. So every time we register new lines, we really see a spike in interest in the lines. So I think it's just really being in people's mind that those resources are available. And we're also very happy to collaborate with scientists in the academic sector, because it's of course great if they publish using our technologies, our materials.